Hey everybody, not often do I do this in the grad class where I make a review video for something, but many of you, almost every single one of you, did express some confusion around gel shifts. And with time in class being so precious this semester because of our hybrid format and the pandemic, I thought I'd put together what will hopefully be a pretty quick review video on gel shifts where we can just take our time and really build this concept to try to make it clearer for all of you. So I'm not going to be introducing anything new here, just talking about gel shifts the same way I did in the lecture, but taking time with the explanation so that hopefully it's more clear. So what I have here on the board so far to orient you is a gel, of course, with 5 wells in it, and then different things that we can add to each reaction mix. In case you can't really read my handwriting here, what we have is the probe, we have the protein that we're studying, we have S-comp, which is the specific competitor, which is unlabeled, NS-comp, the non-specific competitor, which will be unlabeled, and the antibody. And by saying which ones of these we add to each of these lanes, we'll get a better sense of how the gel shift works. So here's an example of the probe. It's a short double-stranded molecule of DNA. I want us to imagine that we're just testing DNA binding of some arbitrary protein. So let's say that that arbitrary protein binds to and recognizes the sequence CCATGA. Not a restriction enzyme, so it doesn't have to be palindromic. It binds to this region specifically. And when we're using a probe here for gel shifts, we make that probe radioactive. So there are phosphorus atoms that are part of this DNA molecule that are radioactive, and they'll expose film. So if we were to run this probe alone, and only the probe, so nothing else has been added, just the probe is in this well, it's a fragment of DNA, so it's going to run to some migration point, pretty low in the gel because it's pretty small. And if we put film over this gel, we'll see a band corresponding to that probe, because this is where the probe bound all by itself. Where the probe ran on the gel all by itself. So now if we repeat the running, but we add the probe and the DNA that we're interested in, we don't add any competitor or antibody, we just add the probe and the DNA. I'm sorry, the probe and the protein. What we can imagine is that if this protein binds to this sequence, then the protein will bind to the sequence. When we add the protein to the reaction mix, we'll form a DNA-protein complex. The protein is binding to its recognition sequence. This complex is a higher molecular weight. It has more mass, more size, more surface area than the probe alone. And so when we run this in the gel, because the protein is bound to that DNA, we're going to get a shift in the gel. That's the gel shift. So this represents the migration of the probe and the protein bound to it, which runs slower in the gel because it's larger in volume and larger in mass. So we get all excited, right? We do this and we say, oh my God, our protein binds to our DNA region. Look how amazing this is. Our protein bound to this DNA region. So then you can begin calling this into question, can't you? So the question first that you might want to answer is, how do you know that it's this recognition sequence that the protein is bound to? Maybe the protein bound to the sequence over here at the 5' end, or the sequence over here at the 3' end. So the next thing you would do is add a specific cold competitor. Now I'll pause the video while I draw this in, because I don't want you to have to sit here while I actually do the drawing, but we'll talk about it once we resume. So what you'll notice in the specific competitor is two things. One, it's unlabeled, so it's cold. We can't see it on the gel. And two, that recognition sequence that we're pretty sure is what our protein is binding to is still there, CCATGAT. It's still present, but the flanking regions have been changed. This DNA is a little bit longer. There's some more sequences in this non-specific competitor, specific competitor. We have different flanking sequences here. So the idea is that if we add this in excess, if we add tons and tons of this in terms of concentration, then our protein is going to preferentially bind to this sequence rather than this one, just because there's so much more of this as target. So to be fair, we are getting a shift when we add the probe, our protein, the specific competitor, and nothing else. We are getting a shift. This cold competitor is actually migrating here on the gel, but it's cold, so we'll never see it. What we only see is probe, and the probe is running down here because it's been swamped out or outcompeted by the excess amount of this cold competitor. So this shows that the protein appears to be binding to our sequence specifically that's somewhere in the middle of the competitor that we're using. So that's a good result. It shows the glimpse of specificity. The next question, though, is how do we know that our protein is binding to this sequence specifically and not just to DNA non-specifically? In other words, if our protein bound to any old DNA, if you were to think of it more like an RNA polymerase than you were to think of it like a restriction enzyme, if it just was attracted to negative DNA, of course it bound to this competitor, because this competitor is DNA and there was a lot of it. This probe was DNA and so there was some of it before. How do we know that this binding is specific? For that, we need a non-specific competitor. Here's our non-specific competitor. Here you'll notice that the sequence is also cold. This is a cold competitor just like before, but now it lacks the consensus sequence that we're sure our protein binds to. If our protein was indeed binding non-specifically, and it just like DNA, and there is a lot more of this competitor than this, we're going to lose the shift again. We would expect this migration pattern when we add the probe, our protein, and the cold non-specific competitor. We would expect this. This would tell us that all of the shifting is invisible to us. In other words, that all of our protein bound to the cold competitor. However, if our hypothesis is correct, and instead our protein binds to this sequence with specificity, that specificity is going to cause our protein to ignore the cold competitor. Since this sequence, CCATGAT, since this sequence is not present in the competitor, our protein will be unable to bind to it. And since our protein is unable to bind to the cold competitor, it will only bind to our probe, and we should restore the shift. So that's a good result. If we restore the shift here, it shows us that that binding was specific, and any old, random, cold, nonspecific competitor can't really give us that loss of the shift. It can't outcompete the protein binding. So that's good. The last thing, like I talk about in the lecture, really gets at how do we know that it's our protein that's binding to the DNA and not some contaminating protein that we might not even know was in there. It's possible that in our protein purification process, we pulled through other proteins we didn't intend to, and those other proteins are here as well, and perhaps one of them is binding specifically to our DNA and not the protein we're interested in. For that, we do an antibody control. What we do is we add an antibody to our protein of interest. And if the other alternative were true, and it was some contaminating protein that was binding there, of course our antibody would not bind because our antibody is specific to the protein we're studying. If the antibody binds, we have an even larger molecular weight structure than we had before. In other words, we now have the probe, our protein, and the antibody. This is much bigger in volume and surface area and in mass, and it should cause a super shift when we have our probe, our protein, and the antibody. A super shift. However, if it was contaminating, then we would have no antibody bound to the complex. This complex would be the same size as before. This would be the result that would indicate a negative result. In other words, that it was a contaminating protein carrying through. But what we expect to see is the super shift. So first lane is the probe alone. It shows us the migration of the probe when it's not bound by protein. Second lane, protein is added. It shows the shift if our protein binds to that DNA. And I said that specifically. If our protein binds to this DNA, we'll get a shift. The question then becomes, did we get the shift because our protein just bound to this DNA in general, or because it bound specifically to our sequence? Well, what we do is we try to outcompete that binding with a different probe that contains different sequences except for the consensus binding site. The binding site is there. And if we lose the shift, that shows us that the specific competitor can bind to our DNA. That suggests specificity of binding. But how do we know that our protein wasn't binding to DNA randomly? In other words, any negative DNA molecule. To address that, we use a non-specific competitor, and we should not see the loss of the shift, or to say it another way, we should see the shift. This shift tells us that our protein is binding to our hot probe, even in the presence of excess nonspecific competitor, because our DNA is essentially ignoring the nonspecific competitor. It can't bind to it because that nonspecific competitor lacks our consensus binding site. And then to determine that it is indeed our protein of interest that's binding to the probe, we would use an antibody to our protein of interest and look for that super shift. Now if we can go back, crudely, I'll admit, but if we can go back to the slide from the lecture, hopefully this makes sense to us once again. So we see the probe runs on the gel by itself. That's where the probe runs. It's low molecular weight for the probe. If we add the probe and the protein, we get our first shift. And that first shift tells us that our protein is binding to the hot probe. And we want to make sure that it's the consensus sequence that's making that happen. So then we're going to add the probe, our protein, and the specific competitor. And when we do that, we add the specific competitor, we lose the shift. Now what this diagram is trying to show is that, yeah, the protein is binding to the specific competitor, but notice that there's no star here. That means that this is not labeled. This is a cold competitor. And so even though the protein is binding to that cold competitor, indeed that's what's happening, we don't see that band here because it's not labeled. All we see is the bottom band, the probe band. Now as we go on, we want to make sure that that binding is specific and not to any random DNA, so we're going to use a non-competitor, a non-specific competitor, and there what we see is we get the shift again, we restore the shift, see there's the labeled probe and it's binding, and down here in purple we see the non-specific competitor. It's running in the gel, it runs to the low molecular weight because our protein isn't binding to it, and we don't see it on the gel because it's not labeled. And then to make sure that it's our protein that's binding to the DNA, we use an antibody that specifically binds to our protein, and we get that super shift, and there's the super shift on the film. So hopefully with a little bit more time spent on this topic, it makes a little bit more sense. Of course, let me know if that's not the case, please, and I'd be happy to meet with you all one-on-one to clarify whatever loose ends might remain.